Hello, everyone. It's my pleasure to introduce Siddhartha Mitra, known to his colleagues as Sid, so easy to remember. Sid has been with us at the Morgan Adams Foundation Pediatric Brain Tumor Research Program now for a year, and we have learned an awful lot from him in this year. He did his PhD at the University of Buffalo and was a postdoc in the University of California, Davis, but has spent most of this decade so far before joining us at the Stanford Ludwig Center for Cancer Stem Cell Research, first as a research associate there, and then as senior scientist and CBIL investigator. Sid really, while he was there, was involved in a lot in the development of primary adult and pediatric brain tumor stem cell lines, and is still, I see, on some of their grants in that respect. Increasingly over his time, though, at Stanford and here, he's really concentrated on a basic science and a combination approach to immunology and immunotherapy in adult and pediatric brain tumors. Without more ado, here's Sid.
Well, thank you, Nick, for that kind introduction, and thank you everybody for coming here for the talk. I'd like to start off by thanking the Morgan Adams Foundation and the wonderful folks who attracted me to come here, especially the great research that has been going on at the Children's Hospital and the Department of Pediatrics. Now, I've got a very unusual title that is Kickstarting the Cancer Immunity Cycle against malignant brain tumors, and I'll spend the first half of, or a little bit of the first half of my talk trying to tell you that it's not a great target, that is pediatric brain tumors are not a great target for immunotherapy, and then I'm going to spend the rest of my talk saying that, hey, maybe we can make it work. But that's also essentially the nature of the beast of cancer, which is always evolving, always tricking us, and we need to find more newer and newer ways to overcome it. Now, to give a very brief summary, I'm going to try and see if this works. Nope, I'll have to stand there also. To give a very brief summary of what brain tumors, or specifically pediatric brain tumors, because that's been one of my focus, but we've also been doing some work in adult brain tumors. Now, one of the first things that I'm going to say is that these are two very different beasts. The mutations are significantly different between the adult and the pediatric tumors. The tumor microenvironment is fairly different. But one thing is common, and that's the myeloid immune system, or the myeloid system, which seems to be predominant in both adults and kids. And that's where the myeloid checkpoint pathway is what my focus of my research has been. Now, most of the kid brain tumors, most of the brain tumors in kids happens in the posterior fossa, and that's one of the major differences between the adults and the kids. But you do get a couple of gliomas which do occur in the frontal lobe. But the big problem is that the kids' tumors, which seem to be embryonic in nature, or also for the longest time were known as embryonal tumors, are rare. They're not as frequent as you see in adult or adult cancers, but they're also very fatal. Now, the standard therapy for pediatric brain tumors, now I really contemplated whether to put in all the big sort of introduction to what brain tumors are. Well, the biggest introduction, or the simplest introduction I can give it, is that if you have a malignant brain tumor, you're dead. That's the introduction I can give you. That's it. So they're bad. Now, what is the standard therapy for pediatric brain tumors? Well, right now, it's mostly surgery, but they do metastasize and then come back, and most of the metastasis occurs in the leptomeningeal space. So you essentially end up giving high-dose brain and spine radiation. That is basically to try and catch if there is anything left behind. And then you go off and start giving intensive chemotherapy. But there's a huge consequence to the survival. Now, the survival rates are good, but once you relapse, there's less than 2% chance of surviving. And the radiation and chemo itself leads to about a 20 to 25% decline in your cognitive ability. So there is a huge cost to that survival that we keep seeing. But now, what are the challenges of treating pediatric brain tumors? Well, everybody will say, first of all, delivery across the blood-brain barrier. Now, I'm going to say that it's not exactly true. There is a blood-brain barrier and there is a blood-tumor barrier. And every blood-tumor barrier is different and depends on the tumor that you're looking at. I talked about trying to preserve survival without any neurocognitive decline. But if you think about it, most chemotherapeutic agents target dividing cells or developmental signaling pathways. And a child's brain, especially at that age, has all of that. It's rapidly dividing, and it still has all the developmental pathways active. So the current generation of chemotherapy mostly tends to target the very same pathways that are required for normal development of the brain. Now, there is immunosuppression going on, means most of these tumors have very low immune infiltrates in them. And I'll talk about this low mutational burden also. But mainly, the inflammation that you get from having a brain tumor or treating a brain tumor with radiation or chemo, you are controlling using steroids, and the steroids themselves impair immune response in immunotherapy. And then there's the last big problem. Well, number one, as I showed you before, very briefly, this is a heterogeneous disease. There is a vast number of different subtypes of subtypes of subtypes in these diseases. All these genomics has basically shown us that medulloblastoma, for example, is now 12 different diseases. ATRTs is 4 different diseases, and they're trying to get that even more. Same way with ependymoma. So there are all these different diseases which encompass or we call as pediatric brain tumors, and we can't have models for all of them, especially in the mouse. And as I said, there are few models for these rare types. So how do we get around trying to study or trying to come up with therapy which can be rapidly used in the clinic? Now, there's one thing which I pointed out was this mutational burden. If you can see, most of the brain tumors tend to lie on the left side of this graph. If you look at this from Schillen's paper in Nature Genetics, you essentially see that all the pediatric tumors tend to be in this lower mutational burden profile. If you look at brain tumors in general, they will probably fall somewhere around here. There are a couple of hypermutated pediatric tumors which have responded to immunotherapy, but I won't get into that. My concern is the ones which are here. So I said they're low mutational burden, very cold. Significant number of them are actually epigenetically driven. For example, ATRT, it's a highly aggressive tumor, very fatal, with just a single deletion in SMARCB1 and SMARCB4. That's it's just a single deletion which drives the tumor, and it's got 4 subtypes from that single deletion. We don't know how, we don't know why, but that's how it is. And then ependymoma, there is a single REL-A fusion. But one thing which I'm going to argue now is that perhaps the new generation, they're all adaptive immune system checkpoint inhibitors. In a child, the adaptive immune system is not fully developed yet. There hasn't seen all the insults, the infections that will give it the vast repertoire that an adult has. Whereas the innate immune system starts developing right in the fetus. And lastly is this low tolerance to runaway immune reactions. So that is basically most of the patients who came off the drug, came off the immune checkpoint inhibitors, had vast inflammation. They just couldn't tolerate the drug because of the inflammation. You're essentially giving them steroids to control the inflammation, and you're just in a cycle where you're not letting the immune system fully attack the tumor. So there's a huge number of problems. So we have to go back to the basics of immunology to try and understand or try and develop better checkpoint or better immunotherapy. And that brings me to the cancer immunity cycle, something that was proposed by Ira Mellman, but I think the idea itself has been around for a while now, that you have the release of cancer cell antigens, which are taken up by antigen-presenting cells, which eventually prime and activate your T cells or present them to the T cells. And these T cells in turn come back and start attacking your tumor cells. Now, this part, the first part, is actually simply dependent on a constant process which is undergoing in homeostasis within the tumor. My idea or my proposal here is actually to see if we can push this system right at this process where we can actually get the antigen or the antigen-presenting cells to eat the tumors, literally eat up the tumors to see if they can have a better presentation or better repertoire of presentation to the T cells. Now, those who are old enough to remember Spock, we always say the T cells are like him. They essentially touch and eliminate, and then you have the B cells, which essentially give out antibodies to go and act like smart missiles and home in onto your tumors. Now, these are the very exciting parts, and then you have your NK cells, which are equally exciting. But I'm going to talk about the macrophages, which were essentially considered as the garbage collectors of your immune system. They would go in and clean out all the dead cells during tissue development, tissue remodeling. But at this point, we can say that they're also very good effector cells which clean out the tumor cells. And we do this using or targeting what is known as CD47, which is a protein which is widely expressed. Now, this is something that I'm going to stress very clearly. CD47 is expressed on every single cell in your body. Normal or tumor, it does not matter. So it is not a cancer-specific antigen. It was identified as a target by its slightly higher expression on leukemic stem cells. It's expressed on red blood cells and prevents them from eating by macrophages in the spleen. And essentially, it transmits a don't eat me signal from the cell to the tumors, from the cell to the macrophage, stopping it. So kind of an immune evasion molecule. So, but how does this work? Well, it requires not only the don't eat me signal to be blocked, which is CD47, but also requires the eat me signal. Now, essentially, what you would have is a tumor cell expressing CD47 binding to SIRP-alpha, which in turn prevents the macrophage from eating it. What we need is the presence of this eat me signal, here simply called as an activating ligand with an activating receptor on the macrophage. So if you block this don't eat me signal, you have the eat me signal initiating phagocytosis. So that's the essence of it. Now, how does it distinguish between a normal cell and tumor cell? Well, most normal cells do not have naturally occurring eat me signals, and the tumor cells do. That is what makes the difference and allows the macrophage from distinguishing between a tumor cell and a normal cell. Now, what does happen? Well, if you throw in macrophages with tumor cells or normal cells and give them a CD47 antibody and you simply make a video out of it, you can see these macrophages seek out tumor cells and essentially eat them up. This can be presented as a phagocytosis index, but I want to get your attention to this part here, which is known as NSC1 and NSC2. These were essentially normal neural stem cells which were derived from human fetal brain and also given to the macrophages to go in and eat, to the macrophages to see if they would get attacked by macrophages when you give them CD47. This was the first paper that we had come out with, and essentially a lot of the data I'm going to present in the same way. We essentially took a patient-derived GBM line, infected it with GFP luciferase, injected it into the forebrain of the mouse, allowed it to engraft, pre-treated and randomized, pre-treatment randomization of the mice based on bioluminescence imaging, and then you start treating the mice, and then you look for tumor growth and survival, etc. And essentially, this is the model that we are looking at. We tried to get the most difficult model that we could. This is just an injection of 1,000 cells on one half of the tumor, or one half of the brain. It crosses the corpus callosum, invades into the contralateral hemisphere. You start treating them with the CD47 antibody, you get a decrease in tumor volume, and you essentially clear out the tumor in this mouse model. We looked at various, so I talked about ATRD, which is I did not talk about ATRD. ATRD is a rhabdoid tumor. We have a couple of lines from these rhabdoid tumors. We have a PNET, a CNS PNET of primitive neuroectodermal tumors, and we did the same experiment with them. We inject the cell, randomize, and initiate treatment. You start imaging the tumor, and you evaluate tumor growth as well as infiltration of macrophages. You see the same thing. There is an increase in survival, decrease in tumor growth in both the ATRTs and the PNET xenografts. We've done this over multiple times in multiple different lines, all patient-derived. We've done this in something known as a DIPG, or a diffuse intrinsic pontine glioma. This is essentially a tumor which till now, once diagnosed, was not even pretty much there. You can't go into the pons. So it arises in the pons, and once you see a tumor there is nothing to do. Now, this is probably the first tumor, and because of its devastating nature, they have actually allowed biopsies to be done for research purposes. So you can well imagine the need for trying to get something or get patient samples to study this tumor. Now, if you inject them, you can see we injected them in the posterior fossa near the pons. You can see the tumor rapidly invading into the entire posterior fossa, actually goes into the forebrain as well. You start treating it with the 47 antibody, you pretty much clear out most of the tumor. So another one of the models that we really wanted to look at was metastatic medulloblastoma. Now, why is it? Because as a neurosurgeon told us, that why are we studying the primary tumor where most of the focus has gone, when you can cut it out and throw it in a bucket in the OR? The patients die from the leptomeningeal metastasis, and that's pretty much like removing butter after you've already spread it on bread. So we tried to develop better models, which we were able to actually get spinal and leptomeningeal metastasis. And that is something which is shown here. This is a c-MYC amplified medulloblastoma. You inject it into the cerebellum, you start getting beautiful spinal metastasis. But not only that, you cut open the tumor, you start seeing leptomeningeal metastasis, which pretty much spreads all over the brain. We started treating it with the 47 antibody, and you get not only the decrease in the spinal met, but you also get a decrease in the primary site of the tumor. We wanted to see if you would do intraventricular administration of 5F9G4, which is the anti-CD47 antibody, whether that will accelerate or give us a better response. So in this case, you have the luciferase expressing medulloblastoma cells injected in the cerebellum, and then we implanted osmotic pumps in these mice, directly infusing the antibody into the lateral ventricle, and then looking at the effect of the antibody onto the tumor site. Now, two things I'm going to point out. First of all, the survival was not as good as giving intraperitoneal injections. So that was, but that's something that has actually been seen in the clinic. A lot of patients a lot of leukemia and lymphoma patients who have gotten Ommaya reservoirs, which are essentially what the neurosurgeons do, is they directly put a reservoir in the brain so you can give the drug to the tumor site. They don't see an effect if the tumor is in the parenchyma. So that is something that has already been seen. But what we did see was there was a rapid clearance of the leptomeningeal metastasis. So if we actually calculated whether or not there was an increase in the effect on the leptomeningeal spine or the forebrain metastasis, it actually worked much better. So this is something we are hoping to actually incorporate into a clinical trial where the primary site is already been removed, and if the patient comes in with a relapsed disease, we can perhaps directly infuse the antibody into the tumor site or the CSF so that we can clear it out. Now, well, one of the big problems in doing xenografts is that we are looking at human-specific reagents on human cells, so we do not get any information about what is the toxicity of the drug on mouse cells. We know that a mouse and a human brain is very different. So how to get over it? Well, if we can do a couple of in vitro assays, and that was where we took normal neural precursor cells, mixed them up with tumor cells, we pre-labeled them with 2 different colored dyes, give them the CD47 antibody, and you clearly see that even when, and we did this at a higher concentration where the number of normal cells was about 10,000-fold higher than the tumor cells, we still saw them preferentially going and eating the tumor cells and not the normal cells. But that's great, that's in vitro. Can we do this in vivo? So what we did was we took normal neural precursors and injected them in the mouse pups, and eventually we actually, using ultrasound, we can go in and directly inject them in the mouse, in the mom, directly into the embryos in the brain so that we can now have a human mouse chimeric brain. And we take a non-transduced medulloblastoma line, and we know that there is 100% penetrance of this tumor line, so we get tumor in 100% of them. But this is now a bed of human cells, which is human normal cells, which is expressing luciferase, and you have tumor cells on top of them. And then we start treating them with the CD47 antibody, and we are just looking at the luciferase signal, which is now coming from the normal cells. And we didn't see any difference, even though we saw an increase in survival. So some of the beautiful data that you get if an undergraduate goes into an animal facility with an iPhone, and that is when you start seeing how do the mice behave. So this is a very qualitative assessment of the mice behavior, those treated with and without the 47 antibody. And clearly you can see the ones which were treated with or which just had control were cachectic, hunched up, did not run around, whereas the one which you give 47 antibody clearly running around are happy, and I think one even bit me at one point. So then you go and see where we have to do it in immune competent models. So this is where we did some adult work, and this was essentially to see whether in a syngeneic mouse model we can get the same effect. And yes, the answer to that question was yes, we do see a slightly better effect. Now this is using a mouse CD47 antibody, which unfortunately did not have the same level of efficacy as an engineered human-specific antibody, but we do see an effect on a syngeneic model. So what's next? Well, we want to see whether we can enhance. So we all know, results in the mouse, we are great mouse oncologists. We can treat mice, cancer in mice for ages now. But we really need to know what will happen in the humans, but we need to have the data to see what is rational combinations. Instead of taking 2 hammers to hit the same nail, we really want to figure out what is the sequence at which you want to do your treatment so that you get the best results. This is not just taking a single, because I always look at it as an activation of immune physiology. This is not just plain immunology, you're activating the immune physiology. So it has to be done at the right time with the right control. So I'm going to give you a few vignettes about some of the things that we have been looking at and going forward with. And one of the combinatorial approaches was with CD40. And I apologize if I jump around too much, so feel free to talk to me after this. I'll be more than happy to. Now, CD40 we know is an enhancer of the T-cell pathway or the adaptive immune system, but it also has a very significant role in the myeloid pathway as well. In fact, it has been known to increase inflammatory macrophages into tumors, especially it was originally shown in non-CNS tumors, especially ovarian cancer. So our idea was that what happens if we get in more macrophages and then treat with 47? A very simple concept here. And in fact, when we did this, one of the things we saw was that if you give CD47, we have seen over and over again, we get this rapid increase of macrophages. Now, I'm going to now switch over from calling them macrophages to F480 positive cells, because when we published the paper, we thought these were macrophages, these were stained with F480. Now we have published another paper saying they may just not be only macrophages, but also microglia, and that would be the talk I'm going to give right after this, a continuation of this talk itself. But one of the more interesting things is that if you do a midway analysis of treating with 47, you have macrophages coming in from the periphery, essentially eating its way from the periphery into the tumors, or inside the tumors. If you give CD40 and CD47 together, now the entire tumor is chock full of macrophages. You see this same thing by, of course, doing a flow cytometry analysis by simply look at the cell percentage of cells which are 45 positive, 11B positive, F480 positive, and you see this massive increase of these tumor-associated or infiltrating myeloid cells when you treat with CD40 and 47 together. And of course, there is a survival benefit when you start treating bigger tumors with them. The second part is that can we increase macrophage phagocytosis by tumor targeting or opsonizing antibody? Now, this is a concept we all know, and this has been around for a while. Almost all the clinical antibodies today, their FC itself functions as an opsonizing agent. That means that it induces phagocytosis to some level or other. What we have is, well, I told you about the CD47 SERP-alpha pathway, and I told you that there are a couple of eat-me signals already on. We have calreticulin and phosphatidylserine functioning as eat-me signals. But we know that there are tumor antigens on some of these tumors. The antibody that targets these tumor antigens has an FC portion that binds to the FC receptor, and this FC receptors itself function as an activating signal. So the concept here was essentially that if we block CD47 and treat them with an opsonizing agent, now can we do a targeted method of phagocytosis and activating phagocytosis using the opsonizing antibody? But the first thing, our biggest problem was for pediatric tumors or pediatric brain tumors and even adults, there aren't many antigens that we can work with, or that aren't many antigens that have been identified. In spite of all the millions of dollars on the TCGA sequencing projects, there weren't any decent or good surface antigens that were identified. So what we have started doing is a high throughput screening of the live cell surface markers. And this is a surfaceome analysis on live cells. Why live cells? To be very clear, dead cells will suck up your antibody and give you false positive all the way around. So we get a patient sample, we separate out the blood and the endothelial cells, we separate out the dead cells, and then only start looking at the live cells, and then scan them with 386 different antibodies to see what is there on the surface. And we have starting to do this in a more throughput way, and hopefully combine it with CyTOF in the future, but CyTOF also tends to not be able to discriminate between live and dead, unless something has changed and I'm not aware of. Not a CyTOF expert, but flow cytometry, this is the best way we could come out with. And using that, we have been able to identify a couple of targets which were validated from previously known previously known immunohistology analysis, but we came out with a couple of good ones, such as B7H3, CD99 in medulloblastoma, B7H3 on medulloblastoma and ATRT. In gliomas, we have seen CD73 in a subset of them, EGFR, and also CD9. And I'm not going to talk about all of them, but the proof of concept was that what is there on the cell surface of all these cells. Now, another thing which I wanted to point out is that this method, we can also multiplex this and throw in normal cells at the same time. What that allows us to do is that we can subtract out any surface expression on normal cells and now only look at the ones which are there on the tumor cells. So it gives us a tumor-specific live cell surfaceome. So B7H3 was something we identified on ATRT. It is expressed on the surface of all ATRT cell lines. And we went ahead and what we have done is we looked at combination phagocytosis as well as in vivo assays of looking at survival to see whether we can not only eat tumor cells better, which the answer is yes, we do that, but it actually also gives a better survival. B7H3 itself is also being now used as a CAR T cell target in a different mechanism, but it was also independently identified by one of my collaborators back at Stanford, Crystal Makel, who is a CAR T expert. So, we also have something, we have the actual clinical trial data now from HU5F9G4, which shows a 52% objective response if you combine 5F9 with rituximab in non-Hodgkin's lymphoma, showing that the proof of concept was we just need to find the right target, especially when it comes to pediatric tumors. Now, I'm going to switch gears a little and talk about microglia. Well, we are all, happy, hey, macrophages go in, they eat your tumor cells. But what about the microglia? They're already there in the brain. Why can't we use them? Well, till now, most of the focus had been on trying to remove myeloid cells from tumors. Everybody said they were bad. Let's remove them. Maybe we'll get a better response. But we are now understanding that they might be used as an effector cell. But our original idea was to show that we really need a peripheral macrophage response to go in and start eating the tumor cells. So we used a mouse model which came out of Richard Ransohoff's lab, and that was basically used in an EAE model of multiple sclerosis, and that was to also sort of lineage trace out the macrophages and the microglia. So essentially use CX3CR1. Now I'll be the first one to say this is not a perfect system, but it allows us to get a better idea, a better handle on peripheral infiltrating monocytes versus brain-resident microglia. So we use CX3CR1. Essentially, you remove one allele and replace that with GFP so that you have green microglia, and then you put them together with another mouse, which is CCR2, which is essentially mostly in macrophages, but is also responsible for the movement of monocytes into tissues. What we did was we took that and back-crossed it into a NODSKIT gamma mouse, which was the more difficult part because we had to make sure we had both the SERP alpha polymorphism so that it could respond to human CD47, but also the knockout of your 3 immune genes. So, yes, it took a while. But it gave us fantastic results. And one of them was to figure out if we engraft pediatric brain tumors into these mice or adult brain tumors into these mice, what is the contribution of the different subsets? So essentially, if you look at the contralateral hemisphere, which is away from the tumor, you mostly see microglia, not so much macrophages. When you add in the tumor, you have both increase in macrophages and microglia on the tumor site. Now, cutting a long story short, specifically because this is this has now been published as of January, the biggest surprise we got was that microglia can also respond in the absence of peripheral blood macrophages. Now, if you do a CCR2 knockout it essentially inhibits peripheral monocytes from entering brain tissue, something that you can see here. There's only a 9% infiltration of this RFP positive population. You treat it with CD47, it doesn't increase. But you do get a decrease in the tumor volume, essentially something that you can see here. Yes, it's not as strong if you have both macrophages and microglia there, but it does work. But are these microglia really killing the tumor cells? How do we really go ahead and do that? So we decided to do live animal 2-photon intracranial imaging. So essentially what we are doing is we're taking the mouse, remove the skull, slap in a window on there, and do 2-photon imaging to see what's going on. Well, what we do is, this is the control. This is the tumor treated with CD47. Green is the microglia, red is the macrophages, and these are the blood vessels. There is tumor right under this imaging, right at this imaging spot, which we have decided not to color so that we can get a better resolution, otherwise things become mixed up. So essentially, okay, so you see these monocytes coming in, they're strolling in the park, don't really go in, but you see the microglia are still there. When you start giving them the CD47 antibody, there is this massive influx of monocytes into the tumor space. It extravasates, crosses the blood vessels into the tumor sites, and starts attaching themselves to the tumor. Now we have higher resolution of this. Essentially, this is a rendering of that entire thing. You can see that the microglia actually throw out these processes and survey the surface of the tumor cell and then start literally eating them up. So the microglia are very active, and when we give them CD47 antibody, they actually have a more precise way of surveilling and eating them. And again, anyone interested, we just published this so you can get all the details from the paper itself. But to me, one of the most interesting part was this phagocytosis at steady state. There is something going on even when you don't give them CD47. This was very difficult to catch, but once we actually imaged this over 3 days, and we were able to catch steady state phagocytosis when you don't give CD47 antibody. You can see the microglia come in and starting to eat the cell, and you will see this rapid, let me go back. You see a microglia right over here. It starts attaching to the tumor and eating it. It's going to the second one, and you'll see one come up here and starting to eat it. So there is this low-level phagocytosis also going on even when there is no CD47 antibody. And this, okay, is this a better version of that? Maybe not. But the most interesting part, or another interesting part about this whole story was that when we isolated the microglia from the macrophages after treating them with 47 antibody, there was no inflammation going on in the microglia. They upregulate their phagocytosis machinery. We are able to designate them perfectly as microglia or macrophages based on their gene expression profile, but they do not undergo any inflammation. In fact, the inflammation or the slight upregulation of some cytokines that we do see is lower than the macrophages when you don't even treat them with 47. So we have a phagocytic cell or an effector cell in the brain which now does not inflame. And so the next part of what my lab hopes to do is to try and identify activators which are microglia specific. And we can think about it simply as a way that, hey, this is probably something that is true because it is a brain resident immune cell and it is adapted to the immune environment and it knows that it should not be inflaming. And that is the next target that we want to go for to see if we can really activate microglia to go and eat tumor cells. And the last part of what I'm going to talk about is this difference between immunogenic cell death and tolerogenic. And this whole brings it to a full circle or a cycle of going back right to the cancer immunity cycle or kickstarting it. Well, the first part I talked about was this release of cancer cell antigens. So you can either, this is an old concept, whether you can do immunogenic cell death or tolerogenic cell death. This is something which has been around for a while. And it turns out, under most conditions, you have something known as non-immunogenic cell death going on and tolerogenic phagocytosis going on, which not only upregulates your T regulatory cells or your Tregs, but also significantly dampens your CTL or cytotoxic T lymphocyte response. And this overall gives rise to tumor growth. Turns out you can actually switch this. A lot of the chemotherapy and radiation agents can actually be used at different doses to be immunogenic in nature, such that you can activate immunogenic phagocytosis and get a better CTL response. And these happen by the use of these damage-associated molecular patterns or eat me signals, which are essentially things like calreticulin, HMGB1, or ATP release, which can go bind to different receptors on your macrophages or your dendritic cells, and in fact push this towards an immunogenic phagocytosis. But how can we get these damage-associated molecular patterns? And we know that radiation itself can induce immunogenicity, and so can chemotherapy. So we tried out, so this was pretty much towards the last part of when I stay at Stanford, and that was trying out these various experiments to see whether we can get a better response. So we combined it with radiation, and we can see that we can get just as well phagocytosis with 2-gray radiation as compared to an 8-gray radiation dose, and something that we have seen where we can actually enhance the survival or decrease tumor growth by combining radiation with 47. But what was more interesting is that what happens when we actually combine it with sublethal doses of chemotherapy. Now, when I say sublethal, specifically we tried to see which are the doses which do not kill the tumor cells, let them live, but can those combine with CD47? And we used a standard CCNU protocol, which is commonly used in pediatric cancers, and we see that sublethal doses of these 3 different chemotherapeutic agents actually enhances phagocytosis. But these are all the old drugs, the drugs that we already know are toxic. Can we go in and identify new ones? So this is something that we recently submitted, and that is, and now, I would say that the drug screening was not to do immunogenic modulators. The original idea for this was simply, this was a collaboration with Mark Remke's lab in Dusseldorf, and that was simply to do a high-throughput drug screen to identify tumor-specific killers or tumor-specific drugs. So what we did was we essentially used, I'm not sure if this is clearly seen, but these are all the different cell lines that we have collected from various patients, and these are a subset of drugs that we screened them with. We originally screened them with 300 different drugs and did an IC50 of every single drug, all the 300 drugs, for every single tumor line that we had. And then we essentially Then we grouped them out together, those which had a very high IC50, so they were of no use because you had to give them at high dose. Then you start looking at those which had low IC50 and those which had IC50 which was higher. So this was for MYC amplified, was one of our major focuses here. So group 1 up here are those which are specific for MYC amplified medulloblastoma. Group 3 here are those which are not for MYC amplified. They are higher, the IC50 is higher for MYC amplified medulloblastoma as compared to other tumors. So these are the 2 different groups. So we are able to throw them out, identified, not surprisingly, a lot of epigenetic modulators, and one of them we homed down to was CI994, specifically because it had a very low IC50 for mostly MYC medulloblastoma and not for the other tumors. This is the data that this is essentially showing what I mean. There was a mean dose response profile, and the validation of screen of CI994 across each entity and subgroup, across all the tumors that we had. And we are able to identify CI994 as something which is specific for MYC medulloblastoma, also seen here. Of course, we went on to do the in vivo study to see what its effect was. But when we did the RNA-seq and methylome analysis, and remember, this is at IC20, not at IC50, this is at IC20 concentration. We did RNA-seq and methylome analysis to see what are the upstream regulators that we can pull out. Our top hits were mostly immunogenic modulators, TLR4, interferon alpha-2, CXCL12, TGF-beta, interferon gamma. So there is this immunogenic, so at low doses you can have an immunogenic induction going on, which allows us. So if you combine that with CD47, you simply do a phagocytosis, you can see a huge induction of phagocytosis in this process. So now we know that there are ways that we can screen for something which comes in at very early. This is something we are hoping to now start doing in primary patient samples. As soon as they come out of the clinic, we want to run the drug screen through the entire panel to see if we can actually identify drugs which are specific for the tumor, but also try to identify better immunogenic modulators for that particular patient. Now, of course, it all comes back to your adaptive checkpoint inhibitors. There are already evidence to show that there is the anti-47 mediated phagocytosis primes an effective anti-tumor T-cell response, both from my old lab and Ming-Michel Zhu's lab, which shows this. But something which really turned out to be fascinating is the timing. So when I did this experiment in two different mouse models, these are spontaneous mouse models of medulloblastoma, one with a p53 mutation and one without. The most fascinating results that I ended up getting was that if you combine CD47 and anti-PD-1 at the same time, you do not get a response, or it's not a better response than 47 alone. The only time we actually got a response was when you give 47 first, give it a 2-week washout, and then start treating with PD-1. So essentially showing that you have to have this priming reaction, and then you start treating with PD-1, you get a better response. And turns out you actually need the 47 SERP-alpha pathway for antigen presentation. So essentially you cannot take two drugs and just throw them in without understanding the biology behind it. When we start understanding the immune system on how it works and how it gets primed and how you can get a better response, you can actually take it to a clinic and define a clinical trial based on that data. In the end, of course, I'd like to thank my old advisor, Irv Weissman at Stanford, and all my collaborators over there, and my wonderful new colleagues, or one-year-old colleagues now, at the Pediatric Neuro-Oncology section here, the Morgan Rabson Foundation, the Andrew McDowell Foundation, ACS, and Alexis Lemonade stand for their support. And I will take any questions at this point. Thank you. No one. Anyone awake? Yes, so the first paper that where we had the adult pediatric brain tumors was done through multiple solid tumors. We have done breast, colon, pancreatic, GISTs, a whole slew of them. I think it's such a sort of conserved mechanism that it seems to have an effect on all of them. In fact, even though the data, actually the second data has also come out in Journal of Clinical Oncology, which talks about all the different tumors. The first result was from non-Hodgkin's lymphoma. The clinical trial also did all the other solid tumors as well. Very good question. I don't know the answer to that. It's been very difficult to get MDSCs out of NSGs. So we haven't been able to, so I have flow plots to see whether they're there or not, but it's not as clear, and we don't know what it's doing. We have taken what we thought were MDSCs and gave them 47, and they seem to be gobbling up tumors just as well, so we didn't know whether these were MDSCs or not. Okay, thank you.